you so much, Kate. And I'd really like to thank you all for, first of all, for being here. It's wonderful to be here in person, I can't tell you. And I'd really like to thank the organizers for this, Brian, Brooke, Mark, Bill, everybody who's helped get this started. So really appreciate being here today. Delighted to talk about our research and updates and things we've learned along the way about genomics in this field of pediatrics. So the title of my talk really, I think, is to me symbolizes the maturity of the field when it comes to sequencing methodologies for pediatric developmental disorders. And I've been privileged to work with a wonderful team of people who have been focusing on a really severe class of disorders that affect children. So I hope to highlight some of this work as we move along. So with that, this is my disclosure statement. I do not have any disclosures to make. So I thought I'd start off really broadly by asking the obvious question that some people may ask, which is, why should we study pediatric epilepsy to begin with? And certainly, and this is potentially preaching to the choir here with our audience, but epilepsy in general is a really common problem. You know, it affects about 65 million people worldwide who currently live with epilepsy, including 3.4 million people in the United States. And certainly, it's a major contributor to pediatric morbidity and mortality. And in fact, most studies show that epilepsy does not exist as a siloed problem or in isolation, and that many individuals who have epilepsy have additional comorbidities, including cognitive deficits, autism, sleep disorders, feeding dysregulation, ADHD, behavioral problems, among many, many, many others. And so it has a really significant impact. And the healthcare of individuals who live with epilepsy is certainly challenging. When looking at the genetics of epilepsy, there are more than 500 syndromic associations if looking in the online Mendelian or OMIM, the OMIM database essentially. So there's just too many syndromes really that are associated with epilepsy. And in part because of that, in addition to other challenges, the diagnosis and medical management of affected children are certainly not uniform. And there's lack of equity across many of our patient populations who live with epilepsy. So in this slide here, what I wanted to show really is a very broad overview of how far we've come when it comes to the genetic methodologies and our genetic workup options for children with epilepsy. Certainly, up until not too long ago, we had very limited genetic testing methods. You know, we would start children with a very basic workup, starting with a chromosomal microarray or karyotypic analysis, for example, with biochemical testing and then single gene testing. And as many of you know, we've come a far way from that. So with the advent of next generation sequencing, we now have the option of offering targeted multi-gene panels in addition to exome sequencing and more recently whole genome sequencing. So the next generation sequencing based tests, which are shown in red here on the slide, are essentially have been revolutionary, basically, for our families, and they've really accelerated the rate of gene discovery for epilepsy. And in this graph that I'm showing on the right-hand corner of the slide, I really like this graph that has been published a few years ago now that actually shows the rate of gene identification in epilepsy and how far along it's moved in the field. So you can see in blue, in what's called the channelopathy era, this was the era that predated next-generation sequencing. So with the standard sequencing methodologies, you can see that genes were getting identified, albeit at a very slow pace. And then, of course, next-generation sequencing came on the scene, and you can see how faster the rate of gene and variant identification became. It essentially shot up in a massively impressive trajectory. And this has been really, really revolutionary and important in both research and clinical care. Now, the focus of our research among all the epilepsy disorders has been on a class of disorders that are specifically structural brain malformations. And in this field in particular, next-generation sequencing has been equally revolutionary. So brain malformations are a really broad spectrum of disorders, and I'm showing here as a kind of a very broad overview the range of disorders that affect the brain that actually have strong epilepsy associations. And these include anywhere from disorders of brain size, where children can have a small brain size, or in some instance a larger brain size, to abnormalities of the cerebral cortex, to abnormalities of the brainstem, cerebellum, and other structures within the brain. So there's a huge class of brain malformations. Traditionally, pre-NGS, these were thought of as distinct entities that are related to an aberration or an abnormality in the very early stages of brain development. what NGS has allowed us to learn is that a lot of the genes, a lot of these disorders are, first of all, actually genetic, which is not something that we appreciated for these disorders. And second is that a lot of the genes are actually linked by cellular pathways, which is what I'm showing you at the periphery of the diagram here. And this knowledge has actually been also revolutionary, because when we think about the cellular networks and the pathways that underlie these disorders, it really puts us in a really opportune position to try to target these disorders via molecularly targeted therapies or drug targets that affect these pathways. And so it puts us at a spot closer to better therapies for some of our children. And this is actually, I would hope, would be something that you would appreciate through some of the disorders that I'll highlight in this talk. Now, my personal story with these disorders really started off actually as a medical student. And I wanted to kind of share with you this journey a little bit, because it started and it ends with a patient, really, with patient care. So as a medical student in a small country called Bahrain, which is shown here in the Persian Gulf. This is a country, by the way, that I personally, I only heard about it when my parents announced to us as a teenager, announced to me that we were moving there. So it was kind of a fun journey. Bahrain is a wonderful and small country in the Persian Gulf. And this is Bahrain. It's an island that's connected to Saudi Arabia via a land bridge. And this is my medical school, the Arabian Gulf University. And in 2003, I met my mentor, Dr. Marie Greeley, who is shown here. So this is really a story of mentorship as well. Dr. Greeley was the sole geneticist at the time in the country. And I heard one talk that she had given at the time, and I'm totally aging myself here by showing you this image of a karyotype. This is how we used to share knowledge at the time, with these very outdated, transparent slides that you put them on the scanner and you project them. And it actually shows a karyotype of a child with Down syndrome. And so this was the one of two lectures in genetics that we got in medical school. And I was inspired by her at the time and asked her that, I told her I'm potentially interested in genetics. What would you recommend? So she facilitated my doing a summer internship at the University of Chicago. So going from the desert to Chicago was a nice experience in the winter. At the time, I did an initial visit in the winter and then the summer following that, which was a little bit better. And at that time, I met up with two additional mentors in my career, Drs. Bill Dobbins, who was at the Seattle Children's Hospital and left last year, and Dr. Ian Glass, who is a professor of genetics at the University of Washington and Seattle Children's, and still is. And at that time, we had medical charts of 5 children who were felt to have a really rare genetic disorder. Their features were a large brain size, or a large head size, abnormalities of the surface of the brain that are shown by the arrow here. Specifically, they had what's called polymicrogyria, which is abnormal gyration of the cortical gyral pattern. A subset of the children had hydrocephalus. And then the other feature was post-axial polydactyly, which is shown on this X-ray over here. So based on these features, we described the syndrome in 2003. We called it MPPH syndrome, named after the 4 core features that I just mentioned. And at that time, we thought that was the end of the story. It's a rare disorder. We've described it clinically to help with identifying and diagnosing children better moving forward, and we thought that was that. At the time, we didn't know the genetics of this condition, nor did we have the resources or the tools to explore it further. Fast forward to 2011, this was at the time when I was at the cusp. I was in the middle of my molecular fellowship and relocated to Seattle, wonderful Seattle. And of course, the UW Genome Science Center here and the University of Washington were essentially the leaders of next-generation sequencing methodologies. So it was extremely fortuitous. So we decided to use next-generation sequencing methods to identify the genetic causes for some of the conditions that we had been studying. And we included children with, a few of the children we had with MPPH syndrome. And we found the genetic cause through these methods. So through exome sequencing of a few children, we found 3 genes that were causal, PIK3R2, cyclin D2, and AKT3. Now, these 3 genes happen to lie in a very important cellular pathway known as the mTOR pathway. For any of you in the developmental biology fields or oncology, this is a pathway that I do not need to introduce. It's an infamous pathway that has really important functions in the cell, not only in development, but also in vasculogenesis, brain development, growth, proliferation, and autophagy. I mean, it's really extremely prolific. And it's beyond the scope of any single talk to discuss the roles that this pathway does, both in health and disease. But it was quite interesting at the time, and we thought this link to the mTOR pathway is potentially interesting. So where have we come since? So we knew that this was an overgrowth disorder, and we knew that this pathway was implicated in normal growth. Our work since then, combined with work from multiple excellent groups across the country and the world, identified that it was actually a spectrum of brain overgrowth disorders that are associated with this pathway. And I'm showing here really this broad spectrum. And it really ranges in its degree and severity from diffuse brain overgrowth, which I'm showing on the right-hand side of the slide, to hemispheric brain involvement, what's known as hemimegalencephaly, where one half of the brain is affected, to much more focal forms of brain overgrowth, including a quarter of the brain, if you will, so quadrantic dysplasia, and focal cortical dysplasia. Now, before these studies were done, each of these were also considered to be distinct clinical disorders. And we now know through the genomic methodologies that these disorders are all linked through the same cellular pathways, and that essentially it's a spectrum of involvement. So this was actually quite fascinating, and it really caused a shift in our understanding. So we went from thinking of these distinct clinical entities as ultra-rare disorders to really taking a step back and seeing that there is a much broader picture over there. So what do we know, really? So we know that there's a spectrum of involvement among children caused by aberrations of this network. And importantly, from a molecular perspective, it wasn't just one or two genes, it was multiple genes that were involved. And it included gain-of-function mutations in activators of the pathway, including, for example, PIK3CA and some of the genes I just mentioned for MPPH syndrome, which are PIK3R2, AKT3, and cyclin D2. And more recently, mTOR itself, the gene mTOR itself was implicated. But there is also another class of functional effects, which are loss-of-function mutations of inhibitors of the pathway. Case in point would be PTEN. And of course, TSC1 and TSC2 are pretty infamous as well. So what was quite interesting is that there was a diversity of causes here. And whether it was a gain-of-function mutation or a loss-of-function mutation, the functional endpoint across these disorders for brain overgrowth were essentially the same, which was activation of the mTOR pathway itself. So we knew that the pathway in these children would go into an overdrive mode and cause the brain to grow at a rapid rate, usually in very early development. So we're talking during pregnancy and early postnatal development. So this was quite important and interesting for us to see, because we knew that if we really wanted to understand these disorders, while we might have to focus on one gene at a time, but the big picture was really, really, really big, essentially. Other insights we've learned along the way were two major insights that I'd like to highlight real briefly. As I mentioned, this pathway is heavily implicated in cancer development and progression. And it's among the most oncogenic, essentially, hub within the cell. So we ask the question, well, how do the mutations in our pediatric early brain overgrowth disorders, how do they compare and contrast to the mutations we see in adults with cancer? So this was something that we tried to really look into a little bit with the tools we had. And these studies, by the way, are by no means complete. I think a lot more analysis and work needs to be done in this space. But in this slide, we ask the question is, how do mutations of one of the key genes, which is PIK3CA, how do they compare between pediatrics with overgrowth and adults with cancer, for example? So in this diagram, I'm showing the functional domains of PIK3CA. In the bottom half of the slide, I'm showing all the mutations that have been seen in PIK3CA in the COSMIC database, or catalog of somatic mutations in cancer, and their distribution across these functional domains. And in the top half of the graph, we overlaid the mutations that have been seen in pediatric overgrowth disorders with a special focus on brain overgrowth disorders. And what I hope you can appreciate is that the mutational landscape looks very similar. It almost looks mirrored, meaning that the most common mutations you see in cancer over here, or what are called the so-called hotspots, and there's about 4 or 5 of them that are distributed here, they're also the most common mutations that are seen in children. And this was quite important because when we talk about targeting these mutations with drugs, for example, using mTOR pathway inhibitors, it's very helpful to see that the mutational landscape is similar with oncology. So this was the first interesting observation. The second observation we made was that actually there were a lot more widely distributed mutations in PIK3CA across the functional domains that were also present in brain overgrowth. And they were present at an appreciable frequency in some of the mutations, like these two that I'm showing over here, for example. So this was quite interesting. When asking the same question with the other genes as well, we noticed a very similar observation. So with the three top genes that we've been working on, PIK3R2, AKT3, and mTOR, I'm showing here the cosmic plots as well, and I apologize if they're small. These are their cosmic plots showing the most common mutations. And the peaks, essentially, that are seen in cancer they also happen to be the most common mutations that we see in children. For example, the G373R mutation in PIK3R2, which is again this peak here in cosmic, it is by far the most common mutation we see in MPPH syndrome. Similarly, with AKT3, this E17K mutation, which is right here, this is also by far the most common AKT3 mutation that is seen in brain overgrowth in children. And similarly also for mTOR, the S2215 FNY mutations that are seen in the kinase domain are the most common mutations as well. So this was quite helpful. The second big observation that I want to highlight is that when assaying multiple regions from the brain, so this relied actually on doing molecular genetics and molecular diagnostic, applying our molecular diagnostic tools on multiple brain regions that have been surgically removed in children who have really severe epilepsy. So typically, a lot of the time, if epilepsy is really severe and intractable, which sadly is a common occurrence in these disorders, a lot of the time, if children fail medical therapies and their epilepsy is not responsive, they undergo resection of that part of the brain. And so we have been through a collaboration with our excellent neurosurgical and neurology teams at Seattle Children's and UW, we've been able to procure sometimes multiple brain regions that have been surgically removed. And when using deep sequencing of these multiple brain regions, what we have noticed is that the level of the mutation in these different regions can vary quite a bit. In this child, for example, so this is one of our beloved patients who had since graduated from high school. We noticed that the level of the mTOR mutation that we identified in him ranged from 9% in the most severely affected region all the way to 0% at the anterior edge of the lesion that was surgically removed. So there was also variability in the level of the mutation or the mutational burden within the same brain in these children. And so this is, of course, a classic example of mosaicism at the tissue level, where we have different fractions of mutant cells in different tissues. When we looked and compared the percentage of mutant cells across these regions with the neuropathology, what was quite interesting was that there was a partial correlation, meaning that some of the regions that had a high mutational load were really dysplastic and had several of the neuropathological abnormalities that we would expect, but it was not a clean correlation. It wasn't perfect, and I'm going to talk a little bit about this later on. And I do think this was actually quite important at the time, because one of the biggest questions we've asked is, does the level of mutation or the percentage of mutant cells determine the phenotype? And that's a really complicated question, it turns out. So I hope to talk about that a little bit more down the line. So to summarize then, what we've learned so far are two big themes here, is that our developmental brain disorders that are associated with overgrowth are mosaic, and this is now my excuse to show you a lot of mosaic paintings or pieces of art, which I tend to find to be quite beautiful, and that they have these developmental brain disorders have extremely strong biological links and molecular links with cancer. And again, this really puts us, I think, in a very good position to try to capitalize on all the advances that our colleagues in oncology have done with not only molecular classification and diagnosis, but also with therapeutics. So with that, in the next part of my talk, I want to highlight two big questions we've asked and share with you some data related to these two big questions. And essentially, they were two summits. Now, as I feel like I'm more of a Pacific Northwesterner, let's talk mountains. So essentially, the first question we asked is, can we reliably detect low-level mosaic variants in children and adults with these disorders. Meaning, how soon can we make a diagnosis? Because as we all know, the earlier we can make a diagnosis, the better it is for affected families. And the second big question, second big summit we wanted to really climb was to ask, are there functional differences between different genes within the mTOR pathway that have therapeutic implications? So, tackling the first question, we understood early on, based on even from studying our first child that I showed you in the earlier slide, we knew that there are several considerations and challenges for molecular diagnosis in these epilepsy disorders, in surgical epilepsy disorders specifically. And the three main challenges, as many of you may know, is first, your sequencing sensitivity. Because the percentage of mutant cells is not 100%, that means we now have to select the appropriate methods that have the appropriate depth of sequencing to allow us to detect extremely low-level mosaic mutations in some instances. The second challenge is also genetic heterogeneity. So unfortunately, going deep often by default means you have to be very specific in your target, right? So that's really an important challenge here. But at the same time, how do we account for the heterogeneity from a genetic perspective? We know that the list of genes that are associated keeps growing, by the way. So it's certainly not just the genes that I showed you. There's been many more that have been identified since. And so how do we find that balance between the depth of sequencing while also the breadth of sequencing? And trying to capture as many of these genes as possible. So that's a really important challenge. And then the third challenge, of course, needless to say, is tissue availability. Obviously, for disorders that are mosaic, that are tissue specific, to the extent we're able to assay or analyze affected tissue, the better we will be to identify these mutations. And most of the time, I think we all like to hold on to our brain cells, as little as I have left. So, I think it's really, it creates a logistical challenge, really, to try to assay brain tissue. And again, I want to really put a shout out here for our collaborative teams, both at Seattle Children's and the University of Washington, without which, this multidisciplinary team, without which we would not be able to really learn as much as we have, because it really takes multiple teams to be able to get these experiments to go. So with that, I want to share with you some data that just recently got accepted into Brain, where we wanted to actually tackle this diagnostic terrain. So we essentially assayed, in our biorepository, we had 58 children who had focal cortical dysplasia or megalencephaly, a broad flavor of brain overgrowth disorders, who underwent epilepsy surgery. We had 124 total brain tissue samples from these children, most of which were fresh frozen, but we had a smaller number of FFPE tissues. Now, in parallel to testing brain tissue, which is the preferred method, we also welcomed any other samples that the families were willing to donate, because with mosaicism, the more samples obviously you have from each child, the better you'll be able to tell what is the range of mosaicism. So we accepted any other samples, essentially, that were available. So it's a minority of non-brain samples that are shown over here, including saliva, blood, skin biopsies, and so on and so forth. Now, this breakdown is actually shown over here, again highlighting that the majority of our samples were brain samples that were surgically removed. In terms of neuropathological diagnoses, so our experimental design was very specific. So we specifically focused on brain overgrowth disorders, including focal cortical dysplasia, which were the majority, hemimegalencephaly, and then we had other malformations of cortical development that are developmental, and other etiologies. We excluded brain tumors, and we excluded any other space-occupying lesions within the brain that were not felt to be genetic to begin with. So there is a circular ascertainment consideration here. We designed a panel called what we call the mTOR hotspot panel. And really the impetus for this is that we wanted to see essentially how low can we go with identifying these mosaic mutations. So the strategy we opted for was to select very few mutations and go very deeply within the tissue to try to find these low-level mosaic mutations. So our mTOR hotspot panel included 6 of the most common mutations or variants that are seen in these disorders in the mTOR pathway, including 3 in PIK3CA, 1 in AKT3, and 2 in mTOR. They are the same variants that I showed you a few slides earlier that are commonly seen in cancer as well. And so to really sequence these variants at a very deep level, the methodology we decided to use was droplet digital PCR. And for many of you, this may be a well-known method. But for those of you who may not be as familiar, droplet digital PCR is one of the newer PCR methodologies whereby your DNA reaction is essentially fractionated into ideally 10 to 20,000 droplets, so containing your DNA molecule. So essentially, it contains really high-depth data from the same DNA molecule or input that you have. The advantages of ddPCR is that it does provide quick results. Essentially, this is a technique that could be run within a few days. It requires, importantly, minimal input of DNA, which is quite helpful when you're dealing with valuable surgical samples, and sometimes post-mortem samples as well. It's more tolerant of DNA degradation. We were able to successfully do it on FFPE tissues, and it does provide ultra-deep coverage. But it is, of course, very site-specific, so it's only ideal for disorders that have a narrow mutational spectrum. You can't do full gene sequencing with ddPCR. Now, using ddPCR, we derived something called the variant allele fraction as a proxy for mosaicism. And what that means is that we essentially compared the number of mutant droplets to the total number of droplets, and we used that to assess the mosaicism burden. Now, of course, as many of you know, this is a sampling consideration. Really, it's not an accurate assessment of mosaicism biologically. It is just a technical value that gives you an idea of what that range could be. And it really depends on which tissue you're testing in the lab. Where's your DNA source from? But it's a good proxy for our purposes. So with that, I'll now share with you a few insights from this experiment. The first insight we learned was that our surgical epilepsy disorders were associated with a wider range of mosaicism than we initially appreciated. So in this graph over here, I'm showing all the individuals on whom we found mutations and their variant allele fractions, or VAFs, shown by tissue type. So brain samples being the predominant are in black. And what we saw is that in some children, it ranged all the way from 0%, for example, as in this child, all the way up to 15%. And again, let's keep in mind that this was from the same tissue that was surgically removed from that child. So that was actually quite a broad range. In other children, it was a little bit narrower, but it was actually quite broad. And in some children, it was up to 20% of cells that were in the samples that we assayed. So this was quite helpful. This is, the range is actually shown here by gene. So what we appreciated is that PIK3CA seemed to have the wider range of mosaicism, whereas mTOR, for example, had a narrow range of mosaicism. Now, these data, particularly because of our smaller number here, are not statistically significant just yet. And certainly doing it on a much broader cohort of ideally, thousands of individuals would be really informative. The other quick observation I want to make here, and this is a question we get all the time, which is, should we test blood from children? Being, of course, the most common approach when we see children in clinic. The yield from blood was 0 in all of our patients. For any of us in the mosaicism field, this is not surprising, of course. Blood is highly clonal, and oftentimes it tends to be of low yield. And so that was actually quite important for us as well. So that means when we see these children in clinic, we usually try to avoid testing blood as much as possible. And with that, I'll show you a few examples to highlight, or a few stories to highlight some of these observations. So this is a child we had seen at Seattle Children's who had hemimegalencephaly, so one half of the brain was really abnormal and dysplastic. She had a vascular malformation that was seen on her leg that was quite significant. Her epilepsy was so severe that her surgery was really life-saving at the time. And using the Megaplex panel that was launched through our colleagues here at the University of Washington, this is our test that was launched many years ago. We identified her mutation to be an AKT3 mutation that was low level and mosaic in form. What was quite interesting is that we sent multiple samples, and some of these, I have to say, were tested in the research lab, and then a couple were tested by the Megaplex panel for practical purposes. But from the biopsy of her vascular lesion, the AKT3 mutation level was actually quite low. It was only seen in 1.3% of cells, in spite of the fact that this is a lesional tissue, right? Whereas in the brain, the level of mosaicism was much more significant, much higher. It was from 10 to 18%. And this is actually, it was quite a lesson for me personally to see is that sometimes it can be really low, and you really need to select, if you're testing a peripheral sample like a skin or blood, you really need to select a very ultra-deep, an ultra-deep method to really be able to find these if you don't have brain tissue or the option of testing brain tissue. And this is, again, our Megaplex panel. This is our lab website that actually shows the panel. It provides really deep targeted next-generation sequencing of all these genes. And we accept a wide variety of samples. We launched it in about 2014, I would say, and we've tested more than 300 patients to date, and it's been, I hope, helpful for our families. So the second patient I wanted to really show and share the story of which is this child who had diffuse brain overgrowth. You can see bilateral brain overgrowth, bilateral brain MRI abnormalities. So this child, unfortunately, because of the bilaterality of his brain findings, he had really severe intractable epilepsy early on, as was not a surgical candidate, and passed away within 10 weeks of life. And the family donated his brain to research, and he had, on autopsy examination, significant brain overgrowth. And his brain weighed a massive amount, 905 grams, which is really, really large for his age. Using the OncoPlex actually platform, we identified an AKT3 mutation on him, which is actually another mutation that's within a key functional domain that was seen in about 38% of cells in his tissue. And this was actually testing that was performed on tissue blocks. Because he passed away suddenly, we couldn't freeze brain tissue, so we had FFPE blocks. And the fact that we were able to perform genome sequencing on that was really helpful. Now, interestingly, what I also want to highlight is that we also found, besides this AKT3 variant, we found two other signals, two other variants that I want to leave this as a scintillating question to the audience. And I welcome really questions and ideas and collaborations to address this, is that using our ddPCR experiment on his FFPE tissue, we also found two PIK3CA mutations on him that were extremely low level. And I'm sorry, this isn't showing very well here, but the VAFs are less than 1% for both of these mutations. And these are actually, are part of our mTOR hotspot panel. And they, by quality, our technical quality criteria, and I'm happy to talk about that offline as well, because we do things in triplicates, we repeat them, we have positive controls, negative controls. We really wanted to make sure, we did the best we could to ensure that this wasn't a false positive, essentially, or an artifact. And they check out. The challenging question is, and again, I leave that open-ended because I don't certainly have the answer, is what are the odds of having 3 mutations in this child? And could it potentially explain his severe phenotype? So that is a question that I personally have. I think this is the last patient-specific example that I wanted to highlight is, it's essentially, it was an educational lesson for me as well, is that really, how do we tackle the diagnostic odyssey when we have children who have mosaic disorders, right? So this is a child who was referred to our research, who had brain overgrowth disorders, phenotypically fit. Everybody really wanted to achieve a molecular diagnosis on her. So we really went through a diagnostic odyssey. So 2011, the family donated saliva to research. We did targeted testing via molecular inversion probes at the time, a methodology that was pioneered by the Shanduri lab. And that was actually high-depth coverage of the target genes, negative. Then the family was extremely keen on a diagnosis. So we got blood and saliva from the child. And we tried to do deep exome sequencing, by the way. So we did 200 to 250x exomes, right? So at the time, I think now it's a little better, but at the time, the gold standard, if you will, was considered like 30x, right, or something, So it was 200 to 250x, certainly on the saliva, negative. We did whole genome sequencing. This was way back when whole genome sequencing was just getting off the ground. This was on blood, and it was 30 to 50x coverage, negative. Finally, a few years down the line, the family agreed to do a skin biopsy. It's something that they had avoided, obviously, because it is invasive, And we did exome sequencing, deeper exome sequencing, if you will, on the skin biopsy piece. And we found an mTOR mutation that was present in 10% of alleles in the skin biopsy sample. And so I just really wanted to highlight this as kind of to me, it was at the time a lesson in terms of how hard sometimes it can be and how valuable it is to assay samples, but at the same time, it's not always doable. And the last observation I wanted to make about this part as well is that we've also identified in these disorders, by the way, several pedigrees where multiple children in the same family, multiple siblings can be affected, suggesting that one of the parents can be mosaic gonadally, right? And we have done deep sequencing of peripheral samples in the parents, not deeper sequencing of gonadal tissues, importantly. And we couldn't pick out these low-level gonadally mosaic mutations in these pedigrees. So this is really another important challenge, I think, is that sometimes these disorders can recur in families, and it can be one of the parents who actually is mosaic. And this actually fits with several other case reports in the literature where they've identified gonadal mosaic pedigrees as well. So to even complicate matters further. Now, stepping back from these specific scenarios, the other observation we made is that also when we looked at the specific neuropathology diagnoses across our cohort, we found that there could be some genotype-phenotype correlations, whereby mutations of PIK3CA appear to be associated with more diffuse brain overgrowth, so either bilateral or hemispheric involvement, whereas mutations of mTOR are usually associated with very focal forms of dysplasia and structural malformations. And this also kind of mirrored the range of mosaicism that I highlighted to you earlier, where PIK3CA appeared to have a little bit of a broader range than mTOR. And the last specific observation from this ddPCR journey that I wanted to show you is that there was some correlation at the gene level, but when we looked at the level of mosaicism and its correlation to the neuropathology, the relationship started to break down, essentially. And this was actually one example of that. And I have to say, much of this work, by the way, again, is I attribute and I acknowledge my collaborators, and I'm simply the mouthpiece for a lot of it. And for this particular workup, for this individual, I really want to put a shout out to Dr. Raj Kapoor from our pathology department, our lab medicine department at Seattle Children's, who did heroic efforts here. So this is a child who had a surgical resection of the temporal lobe, and we had two broad regions that were resected, superior temporal and inferior temporal, for his epilepsy. And we got multiple subsections within these two broad regions, so 2 pieces and then 4 pieces from the inferior temporal, and we subdissected them at the time into several individual pieces. And what we found at the time, and this is, by the way, pretty standard in our neuropathology, through our neuropathology workup, it's part of the diagnostic workup, is that they would dissect the regions and perform their neuropathological examination. And we found a PIK3CA mutation, one of the hotspots, at an extremely low level. This is less than 1%. And it was only seen in 2 pieces within the inferior temporal region, whereas all the other pieces were 0%. And again, we went, by the way, on a really extensive journey there to technically confirm this, because it really falls within that really tricky range where it could be a false positive. So happy to talk about that later if there's interest. So then we ask the question, okay, can we dig deeper and see if we do a more microdissection of these sections and actually do our ddPCR test just for this PIK3CA mutation, is there a correlation between the degree of neuropathology and the level of mosaicism? And the broad summary, I really, unfortunately, don't have the time to go through the individual details of all of this, but the big observation that we made, and I hope this can be seen over here, is that essentially there was no correlation between the level of mosaicism or the mutant sections and the non-mutant sections, because in some of the superior temporal regions, for example, where we found 0% level of mutation, they had really dysplastic regions with very large dysplastic neurons, what's called balloon cells, which are characteristic of these disorders. These are actually phospho-S6 positively staining cells, which, as many of you may know, phospho-S6 is a downstream marker of the mTOR pathway. It's a marker of mTOR pathway hyperactivation. And you can see these large, very positively staining phospho-S6 cells. And other regions that are also non-mutant had more normal morphology. And similarly within the inferior temporal, by the way, where we were able to validate the mutation in our microdissection experiments, we also saw similarly this broad range. So there was no direct correlation between level of mosaicism and neuropathology. And that raises a few hypotheses and interesting questions that I'd be happy to talk about as well. And then the last question we then asked is, okay, if the levels of mosaicism do not correlate with neuropathology, is there a difference in other phenotypic aspects? And the main question we asked was, what about the seizure onset? Is that different depending on the mutation? And what was really interesting is that we found that children who had one of these hotspot panels had an earlier onset of epilepsy than children who did not have one of these hotspot panels. And this is shown over here as a group. And that difference was statistically significant. And the average age of onset for the kids who had one of these mutations was about 12.7 months, whereas the kids who did not have one of these mutations was much later. And this was actually quite helpful, is that then these genetic findings really had an impact on their clinical counseling as well. So then the second question we wanted to ask, and this is the last part of my talk, and I sort of apologize ahead of time because this is data that is really much more complicated and nuanced than I'm going to show you, but I really wanted to highlight this to you because I think this is where the future, hopefully our future work will be, are there functional differences between these genes that impact treatment? And the reason we ask that is because, as many of you may know, there are a lot of mTOR pathway inhibitors out there that have been used largely in oncology. And case in point would be the infamous rapamycin or everolimus, which is an mTOR inhibitor. But there are certainly many other inhibitors and dual inhibitors as well. And so the question that was asked and continues to be asked is, if children continue to have epilepsy, should we treat them? Should we put them on an mTOR inhibitor? But before we get to that question, before tackling it, what we felt is, well, we first need to understand the mechanisms of epilepsy in these children, right? And this is actually work that I think the field of tuberous sclerosis has really paved the way for us, because that's something that's been addressed and continues to be worked on for tuberous sclerosis. And I think the same situation lies here, is that there are multiple mechanisms by which this pathway can cause epileptogenesis. It includes abnormal circuitry. It includes abnormalities of the cells themselves, including not only abnormalities in proliferation, it could be abnormalities in cell death, inflammation or neuroinflammation. It could be actually abnormalities in the molecules, channel function, for example. So there's a pleiotropy of mechanisms here. And so we wanted to really purify this question a little bit. And we wanted to ask at a very simple level, can we at least try to scratch the surface using some human-derived in vitro models, which we felt were appropriate because we wanted to ask things using patient-derived cell lines that we had in our lab. So we used a tried and true in vitro experimental paradigm, which is to take skin biopsies from children who have these disorders, who we had genotyped, and then we would take, of course, healthy donors, if you will. We used CRISPR-Cas genome editing to either correct the mutation in the mutant lines or insert it into the non-mutant line, and then we generated neuronal cells at different developmental time points, starting from iPSCs all the way to cortical neurons, and then lastly, 3D brain organoids. And so the two cell lines we chose, we really wanted to simplify. Unfortunately, this is really a massive amount of work, and it takes really many years of a person's lifetime to do. So we wanted to ask a very superficial question at the beginning. Are there differences between a gene that is upstream in the pathway, and let's select PIK3CA, and a gene that is downstream in the pathway, and we selected mTOR. So we wanted to use two genetic nodes as a handle to start understanding, are there similarities or differences between them in vitro? So these are the two cell lines that we selected and the two patients that we selected. Our PIK3CA mutant cell line came from a male who had actually somatic overgrowth. And so those fibroblasts were mutant, and we identified his PIK3CA mutation at a VAF level of 22 to 24%. And he underwent debulking surgery of this macrodactylus toe, or toes. And then our mTOR mutation came from a child that I had seen in the clinic at Seattle Children's who had diffuse brain overgrowth and autism and somatic overgrowth, and she had this mTOR mutation that we identified in her skin. So we had these two fibroblast cell lines, and we used them for all the experiments that I'm going to show in the next 3 slides. The first thing we appreciated was that at the neuroprogenitor cell level, our mutant NPCs were both hyperplastic, so there was more in number compared to controls, and they were also larger in size. And that's actually an interesting question, because that's one of the also early questions we have gotten. It was, what is the mechanism of brain overgrowth? Is it more cells there, more neurons, or is it actually that the number of cells is normal, but they're larger in size? And what we found was that it's a little bit of both, which is interesting. And this is actually shown here through, so each of our cell lines are shown in a different color compared to controls, which are in blue. And then we have PIK3CA in red and mTOR, both heterozygous, and we generated, by CRISPR, you sometimes get different combinations. We got a homozygous as well, which is not very meaningful clinically because we don't see it in the homozygous form in children. But we nonetheless assayed it. And we found that they were duplicating much faster in culture, so there were larger numbers of them in the mutant lines. So that was actually, and these were all statistically significant. When we looked at the size, we actually assayed both nucleus and cytoplasm. And we saw that they were larger in both compared to controls. And interestingly, PIK3CA was also larger than mTOR. So that was actually quite interesting. For the organoids, unfortunately, our organoid work, organoids are essentially like, like children. We have to take care of them in the lab. They're really high maintenance. They take a really long time. Ideally, you want to grow them for years. And our work here got interrupted because of the pandemic. So we had grown them for only a short period of time of a few weeks. But our organoids actually, at the time, interestingly, they showed our PIK3CA and mTOR mutant organoids. I hope you can appreciate this, and this was really helpful to see. They recapitulated the brain overgrowth phenomena in a dish. They were both larger than the controls, and significantly so. Interestingly, also when looking at the differences between PIK3CA and mTOR, PIK3CA appeared uniformly large, whereas mTOR appeared to have a lot of dysplasia, a lot of things going on at the surface of this organoid. So this suggested that there could be some differences between them. So that was it was really quite helpful to see that they recapitulated the phenotype. And so this is a really busy summary, and I do apologize, but essentially I wanted to just really summarize it and say that I think what these experiments showed us is that using our in vitro tools from patient-derived cell lines in comparison to controls, we were able to discern that there are multiple abnormalities at multiple developmental time points in these disorders, multiple developmental time points in brain development. And that there are potential differences between mTOR and PIK3CA, and they're not behaving the same. And this is really, I think, the promise of precision-based medicine, right? So that we incorporate the genetic information. And the reason why that's valuable, I think, right, is because now we have drug targets, that could be different. So we selected then 3 drugs that target 3 different nodes, starting with everolimus or sirolimus, which targets mTOR. And then we selected 2 other inhibitors, an AKT inhibitor called AKT inhibitor 4, and the PIK3CA inhibitor called alpelisib. Now, many of these, not all of these, but many of these are FDA approved for a variety of indications. But the effect of neuronal growth for many of these is not really well understood. And importantly, actually, for many of you who may know, alpelisib or Piqray, also known as Piqray, recently has had a lot of attention in the media because it is now FDA approved for PIK3CA positive breast cancer in adults. So our phones were going off the hooks at the time because our families were asking whether or not we could use this drug, especially for children who continue to have epilepsy. And in this last data slide, I promise, what we did was that we then used these drugs on our cell lines in the lab to see, are there differences between them? And what we did was first use everolimus as kind of a control, right? Because everolimus, there's a lot of data about it, including in brain cells. And so we kind of used it as our benchmark. And we noticed that everolimus did rescue, at least through our limited assays, it rescued the phospho-S6 hyperactivation that we saw in our cell lines. And that was actually quite helpful. And by the way, this was only done on PIK3CA for now. We're still in the process of doing some of these experiments. But in our PIK3CA mutant cell line, everolimus rescued or decreased the level of phospho-S6 hyperactivation compared to controls at different dosages, which was quite helpful. Alpelisib, which is Piqray, was actually quite interesting to use in the lab because, again, there have been, to my knowledge, limited studies on the use of alpelisib for the brain. I think this is confounded by the fact that the blood-brain barrier penetration is not expected to be good. But needless to say, it was quite interesting to use because what we noticed is that alpelisib actually did reduce, I should say actually probably the appropriate observation here is to say reduce, not necessarily rescue, but it reduced at the neuroprogenitor cell level the hypertrophy that we saw. And this was an effect that was specific to PIK3CA mutant cell, the mutant cell line, not the mTOR mutant cell line. Now that might not be surprising given that it's a PIK3CA inhibitor, but that's really valuable, I think, because again, if we want to treat our children, it does suggest that genotyping them does affect or impact the drug selection or the appropriate drug that we would prioritize. So with that, I really wanted to come back to this and summarize and say, we've come a long way, and certainly now we have awesome genetic tools available to us, but our families with surgical epilepsy disorders have additional challenges, which are the fact that a lot of these disorders are mosaic. So while we continue to talk about omics in the era of, everything is omics, right? Here we're also talking about additional layers, which are tissue-specific omics, the need for ultra-deep sequencing, and brain tissue mosaicism detection. And what we're hoping to do now in the next phase of our work is really bring this all back to the clinical field and try to build this into their clinical workup. So We have, of course, our clinics where we try to now offer them pre-test counseling, offer testing clinically through Megaplex and other approaches. And then, of course, the interesting challenge that I really hope we work on together is counseling and incorporating this into their clinical care, including eligibility for clinical trials moving forward. And really, I think this fits into this broad clinical translational wheel that I think we all think about sometimes, which is how do we take things from the bedside to the bench and back, as they say. And for these disorders, again, I have been privileged to work at our awesome institutions in Seattle that I think collectively across we have so much expertise in town. And I just highlighted one little corner of this, by the way. There's a lot of amazing work that's being done on the natural history side, in preclinical models and basic functional models, as well as more clinical trial work that I do not have the opportunity to highlight. And with that, I'll wrap up and thank the many, many individuals without whom this work would not be doable, including our pediatric neurology and neurosurgical teams, our neuropathology team and lab medicine teams at both institutions who've been just amazing colleagues to work with, my lab, who are really an amazing group of people who are extremely passionate, and it's always for us a point of celebration when we find mutations in the lab, and we're extremely passionate about trying to solve these mysteries and challenges for our children. And then, of course, our funding sources, including both at Seattle Children's, University of Washington, and the Brockman Beatty Institute. And with that, I'll wrap up, and I thank you for your attention, and I'm happy to answer any questions. Thank you.
Can I ask about you're presenting some of the data with the mTOR, I think it's specific, the mTOR negative mutations with a later onset.
Yeah.
So what do you think about the ones that were mutation negative but show an earlier onset? Do you think you're just not finding the mutations and it's a matter of sequencing more and more samples or looking?
Yeah, that's a great question, Kate. So the question, to reiterate real quickly, just in case for the recording, is that for the mTOR mutant positive versus negative children, what are the possible causes for the mTOR mutant negative children? So there are a couple of considerations we've entertained here. Is that it's possible that there are lower level mosaic mutations that we might just miss, in our method of detection, or we've had a lab error, for example, that we might have missed them. The second, actually more compelling, to be honest with you, my hypothesis that I think is that it's possibly that it's other variants either in the same genes or in other genes, because our method is so site-specific. And this is an important caveat, by the way. I want to disclose that perfectly, is that we haven't really looked broadly across the rest of the genes or other genes, and we know now that there are many more genes that are identified. But that's kind of been like the challenge, to be honest, for feasibility, is how broad do we go? So that's the other possibility there, is that they could have variants in other genes. The other possibility that I think the field is also entertaining is what is that interplay between genetic and non-genetic factors for these kids, because certainly some of the children could have vascular or acquired insults or injuries, and those could always be implicated. Those are really hard to quantify, but they can also be implicated for some of these children, and that's something we haven't really looked at yet. So, yes.
I was just wondering, what's the earliest you've ever been able to diagnose the mutants in the genes? Because I know you said you couldn't really see any correlation with the parents. So like at what age or at what point are you able to finally get like positive readings on the mutation?
Yeah, I hope I'm understanding your question correctly. So the question is what is the earliest time point during which we were able to pick up these mutations? So the broad the answer, I feel, is that, first of all, I think that these mutations can happen anytime in development, anytime from as the earliest stages to later stages. And the reason I say that, by the way, is because even though most of our work was on children with mosaic mutations, which suggests that their mutations originated later on in their development, and so it affects a fraction of their cells in the brain, we have also children who have germline mutations that are present in 100% of their cells. So it suggests that their mutation happened extremely early on. So I think that it can happen at any time during development. For our kids with the mosaic disorders, it gets back to their clinical workup, I think, a little bit. So because typically they present with epilepsy, and their epilepsy can be as early as day of life 1. The typical, I'm not a neurologist, but the typical workup plan is usually to start them on standard medical anti-epileptics, so the standard anti-epilepsy drugs. And then if they fail to respond to those drugs, and usually medical intractability is sort of defined as being on multiple anti-epileptic drugs at really high doses, and then if they don't respond, then usually they're put in on the surgical path to see if surgery would be the most appropriate approach. And then from there, of course, that's when we are able to do our molecular workup. So by that time, usually the earliest ages I've seen have been a few months of age. For the really severe children, they would have gone through this pipeline within a few months. Sometimes it's a little shorter even, like if it's really severe epilepsy. And certainly for hemimegalencephaly, where like the brain is clearly abnormal and dysplastic, sometimes they're put on the fast track to undergo epilepsy surgery because unfortunately their epilepsy is going to be really hard to control medically. So that's usually the earliest age. The latest age, to be honest, is kind of something that we are now trying to tackle. We are now extending our studies to adults to try to see if we can find the same mutations affecting adults who undergo epilepsy surgery, because that's something I think it hasn't been known just yet. I think some of it is for obvious reasons, is because a lot of the time the adults might actually be diagnosed or undergo surgery when they're younger with these disorders, but we're trying to actually do the same methods at later ages. I hope that answers your question. Yeah, hi, Brian.
Questions online. So one from Colin Pritchard. So it says, great talk and amazing work related. Do you know of any kids with mosaic Victory 2A who have taken epilepsy? I'm not sure that. Thank you. And if so, what was the experience with them?
Yeah, that's a great question. This is the most active question right now. So thank you, Colin, for that question. So yes, so kids with overgrowth disorders, pediatric overgrowth disorders, not brain involvement, there's a lot of active work that's taking place with putting them on alpelisib right now if they fail other standard methods, including at our vascular anomalies team at Seattle Children's. There's already a number of children that are on it. Those are the children who have overgrowth, somatic overgrowth and vascular and lymphatic malformations. And a lot of the time, their therapeutic endpoint, if you will, the goal of the therapy is to try to reduce that growth and use volumetric images. Now, with the brain disorders and epilepsy specifically, which is where we've been in, we're a little behind with understanding this, and I think it's been hard to do this because of some of the pharmacokinetic and pharmacodynamic data about alpelisib and its blood-brain barrier penetration, and limited data about its effects on brain function. So just in the past few weeks, to be honest, I've heard of a few children, not many to my knowledge, who have brain overgrowth, who are getting started under compassionate care use, are getting started on Piqray, or alpelisib, I call it Piqray just to make it easier for me to say, to treat epilepsy in a couple. For others, it was neurodevelopmental endpoints. It's a little bit variable in terms of the goals of therapy, but a few children are getting placed on it. Clinical trials have yet to come for the CNS features or CNS symptoms yet. But for overgrowth, clinical trials are on the way. So, yeah. Yes, Brendan.
Another question from online. So, Jung Hurd asked, I'm certain it was considered, how about testing on CSF?
Another great question. So yes, absolutely. So first, two quick things to summarize this exciting, future direction is that cell-free DNA from CSF, so specifically cell-free DNA from CSF, has been recently shown, and actually our lab medicine team here at the University of Washington is the leading, among the leading edge of this more than I am. But cell-free DNA from CSF has been shown to have a utility for molecular diagnostics in brain tumors, and more recently in brain malformations, including these brain malformations. So we know that technically it's possible to use to test cell-free DNA from CSF to pick out mutations. So far, there have been 3 papers about this for brain malformations specifically, where the cellular burden was high enough, the mutation burden was high enough to be detected. So I think all of these were high-level mosaic mutations, if you will. They weren't really in the realm of the really low, less than 1%. So we are actually in the process right now. We have a project that we've designed with our neurosurgical team to do cell-free DNA analysis from CSF. We started actually right now with doing it from a dural CSF sample when they go to surgery as a pilot phase. And if we are successful, and this is work that's led by one of our fellows, Wei Liang, from one of our fellows in genetics, if it's successful, the promise would be is that could we do a lumbar puncture, on children and find these mutations? I personally think, I'm very optimistic about this, but I do think, to be unbiased as well, I do think that the biggest challenge we will have is mosaicism and the yield of DNA from CSF, which I think can be low, and whether we will be able to technically find these mutations. Oh, there's Tina. There's the master right there. So so I do think that this is there are definitely going to be some challenges, I think, but I think the data so far has been promising. So yeah.
Excellent. Well, thank you again.
You're welcome.
We'll talk.
Thank you so much for your attention. Thanks.